that my name is Dr. Adejoke Elizabeth Memudu, and I am an anatomist and a neuroscientist in the Department of Anatomy, College of Medical Sciences of Edo State University in Nigeria. And my research work has focused on studying on memory disorder, particularly creating models to study memory disorder like dementia, Alzheimer's disease. And in the course of my studies, we have been able to understand the neuropathology of this memory disorder, as well as the role of astrocytes and neurons as a potential therapeutic agent to avert or reverse the neurodegenerative process occurring in memory disorder that will cause impairment in neurocognition and leads to loss of memory. Then in the course of my studies, I found out that there's a correlation between antidepressive depressive disorder or mood disorder with memory disorder. So that was what brought my study, talking about the antidepressant-like activity of ketamine in chronic mind stress model and experimental study. And before I dive into the study, I would like to establish the fact that memory and mental disorders are related. How are they related? They are related that both are mediated by oxidative stress. And we know that oxidative stress is the major stress that results in neurodegeneration and neurological disorder that cause loss of neuron due to loss of neurite or synapses that result in cognitive decline, loss of memory, and also lead to depression. Then major depressive disorder is being reported as being the fourth leading cause of health problem, and it is characterized by anhedonia, poor eating habit, neurocognitive dysfunctions pertaining to decision-making and memory. So there's no way we will talk about memory loss without talking about depressive disorder, because studies have also established that people living with Alzheimer's have an association with depressive disorder. It's also been reported that aged people diagnosed with memory disorder, there's also a correlation that some of them now get into the process having mood disorder. Now, so depression also has a significant impact on neurocognition and memory. So based on that fact, we also found out that the World Health Organization has reported that depressive disorder is expected to increase. So if depressive disorder is increasing, meaning individuals with depressive disorder will also have an underlying disorder which has to do with their loss of memory. And it has become the second contributor to global burden by 2030, from an epidemiology study done by the World Health Organization. And it is essential to identify new therapies for managing and combating depression that will now lead to alleviation of memory loss in an individual. But we need to establish that there are drugs that have been used to combat depression. And most of these drugs has to do with serotonin reuptake inhibitors. Serotonin is an happy hormone. Serotonin is an hormone that makes you to be happy, aside from we have dopamine. But because this serotonin affects mood, so over time, the therapeutic for depressive disorder or mood disorder has been selective serotonin reuptake inhibitors that works on monoaminergic system. But this fluoxetine, which is the drug that works on monoaminergic system, has been reported to have delayed response time. So it's not really giving the expected outcome needed by clinicians. So reports have shown that there's a need to discover new therapeutic that will be fast acting. It's not that we don't have therapeutic, but we need a fast acting therapeutic that will ameliorate depressive disorder and also end up ameliorating the side effects which has to do with memory. Now, we have various studies that now found out that, oh, glutamate is an important neurotransmitter that works on the glutaminergic system. And this system helps to improve memory and also alleviate mood symptoms. So this glutamate is particular on a receptor, which is the glutamate N-methyl D-aspartate receptor blocker, which is ketamine. And it has been theorized that it has antidepressant property. So that's the journey that leads to the discovery that, okay, ketamine has a significant role in averting depression. But as this research is going on, people are now linking from clinical perspective, like memory deficit is linked to functional disconnectivity in major depressive disorder. Hence, can depression cause memory loss? Memory deficit is linked to functional disconnectivity. What is functional disconnectivity? Functional disconnectivity has to do with loss of synaptic communication between the neurons. The neurons are not communicating from the presynaptic to the postsynaptic region, hence causing mood disorder. And this is also related to memory loss. And it has been established that actually there's a correlation from this question. Can depression cause memory loss? Is there a link between memory deficit to functional disconnectivity in major depressive disorder? Yes, there are a lot of reports that there's a correlation between dementia or memory loss and depressive disorder. And according to Yall et al. 2020 and Lancet et al. 2020, he made a report from his own study that dementia or memory loss or Alzheimer's disorder has been associated with the presence of depression. And also established in their report, there was a discourse on age individual. Age individual, there's a connection between aging and memory loss. And they also find out that individuals with memory loss in that age bracket also have a symptoms of mood disorder. So what is this correlation between dementia and loss? The correlation between them is their neuropathological changes in the brain. And these neuropathological changes is linked with structural changes in the neuron, as well as neurochemical deficit, talking about the neurochemistry of the in the synapses, talking about the action neurotransmitters involved. So all these deficits result in major depressive disorder that causes neurocognitive impairment. This neurocognitive impairment is characterized by recurrent episode of sadness, anhedonia, reduced concentration, recurrent suicidal ideation, and the memory alteration, which is what has been established. But we can see now for in this diagram, this individual, for us to correlate this effect, we can't be extrapolating all this study as per neuropathology in an individual, which is basically done using an MRI. But we start by using animal models that correlate to what is happening in the individual human. So depressed mood cause an inhibition in the psychomotor of an individual. This inhibition is now bringing behavioral changes we see as anhedonia. Anhedonia means loss of interest in what an individual loves to do in time past. It also causes to helplessness. Helplessness means loss of hope. Someone is not like despair, behavioral despair, coping deficit, cognitive impairment that has to do with decision making, thinking, and memory, sleep disturbances, vegetative symptoms, and weight change. This weight change has to do with diet and eating habits. So to further establish it, another study by Lee et al. 2018 now mentioned that the rate of depressive disorder is higher in dementia patients, Alzheimer's disease patients, and vascular dementia patients, meaning all these patients with memory disorder also have depressive disorder in them. as compared to LD control in a clinical study. So it has been established that depression can affect cognitive function, and neurocognitive function has to do with working memory, long-term memory, and all this stuff can cause this cognitive impairment, which is the core part of depression. And you know, an individual with a depressed individual has this type of loss of memory, which is the autobiographic memory, which is the person is losing its individual self-recognition. So I've established that the link is associated with neurodegeneration. Neurodegeneration means loss of neuron integrity. The neurons carrying the neurotransmitters that meet at synaptic cleft to send information for happiness, neurocognition, memory potentiation has been degenerated due to stress. Psychological stress is one of the underlying factors that cause neurodegeneration in Alzheimer's disease, talking about memory disorder, as well as mental disorder or mood disorder, which is depression. So this degeneration causes disruption of the functioning of the brain, talking about the functions of the neurons in the brain, function of the neurotransmitter release that control mood regulation and leads to manifestation of these symptoms of depression, as well as symptoms of memory loss. Hence, this is the major cause and risk for developing depression in Alzheimer's disease, in memory disorder. So what are the areas of the brain involved in this memory disorder? These areas of the brain involved in memory disorder as well as depressive disorder are similar. These areas are hippocampus, prefrontal cortex, and amygdala. The hippocampus is the center for memory potentiation. Prefrontal cortex is the area for neurocognitive function, like executive function, planning, reasoning, choice making, and also has to do with this memory, decision making as per memory. While the amygdala has to do with memory pertaining to emotion, sad emotion. Now, the 5 causes of memory loss needs to be established aside from depression. We have other causes like Alzheimer's disease I've mentioned there, minor or traumatic brain injury, brain tumor, vitamin C deficiency, and alcoholism. These also have a part in triggering memory loss as well as depression. As we mentioned, chronic stress, all this stuff, diseases came to be because of stress, physiological stress, psychological stress, all cause neurodegeneration in the hippocampus, the amygdala, or prefrontal cortex that leads to these changes in the neuroconnective pathway that will now dispose individuals to having memory loss, okay, or depressive mood. Now, so there's this diagram here is showing you that there's a connectivity about memory loss between depression, and anxiety. So all these two, anxiety has to do with neurodegeneration and synapses, loss of synaptic connection. Depression has to do with neurodegeneration and loss of synaptic connection. And hence also both of them have something to do with memory loss. So all these things I mentioned earlier about the psychomotor function of the neurotransmitter that has to do with improving memory has to do with glutamate, which has been sent as the alternative drug aside from serotonin with fast action that can be helped to alleviate this stress-mediated memory loss or stress-mediated or depressive events by increasing, either it is done by either increasing the release of glutamate in the synaptic cleft or reducing its reuptake in the synaptic cleft, so that we have enough glutamate within the synaptic cleft that is activating the neuron and creating opportunity for the memory and the neuron activities to be effective. So if there's an impairment in this pathway, it leads to anhedonia, loss of interest that we've discussed, that I mentioned earlier, leading to depressive disorder. So there are two cells in the brain that are a very important role in normal functioning of the neurons in neurocognitive activity. And these cells are astrocytes, which is a supporting cells. And we have different types of astrocytes: protoplasmic astrocytes, fibrous astrocytes. And these astrocytes, they are just like supporting cells. And they are being implicated to play a key role in depression and Alzheimer's disease, which is a type of memory disorder. There's another stuff which is a protein, which is a synaptophysin protein. It also has an important role in memory and mental disorder because it is involved in all those synaptic connections. So this protein, both of them, astrocyte is a supporting cell, but it can be immunohistochemically demonstrated using the glial fibrillary acidic protein immunohistochemistry to demonstrate this protein, which is basically like a microtubule. That's like the protein from GFAP. Synaptophysin is also a protein found within the neuropil, demonstrating presence of synaptic connection. So there's a correlation between activation of these astrocytes and expression of synaptophysin that leads to learning. If astrocyte is activated in neuropathology of stress, a neuropathology condition induced by psychological stress, the astrocyte respond to protect the neuron from stress or cell death by activating expression of astrocyte. This activation of this astrocyte in the process of in disease state will cause a decrease in expression of synaptophysin. Why? The neurons are already, expression of astrocyte showing is an indication of neuroinflammation, neurodegeneration. So meaning there's a loss of neurites and synaptic connection, causing a decrease in synaptophysin. And also this one leads to loss of memory. We can see the correlation here for elder brain and aged brain. The aged brain can be used to represent like an Alzheimer's disease or a memory impaired disorder, where you see the astrocyte being supportive of the neuron involved in repair function, and the neurons are now protected for their normal synaptic connection and information processing. But in the process of inflammation, you see this astrocyte responding, being reactive, trying to mop out the debris. That's why you see them being reactive. Then in the course of this neuroinflammation, in Alzheimer's disease, you see them contributing to depositing amyloid beta plaque or the tau tangling that causes neurodegeneration, loss of the neurons, and affects causing a decline in neuroprotection, increasing inflammatory mediators. So in essence, astrocyte neuron interaction is significant in controlling neural circuits involving emotion, learning, and memory. Hence, they have been implicated in psychosocial and cognitive impairment. Psychosocial has to do with depression, cognitive has to do with memory, Alzheimer's disease. So these two plays an important role in mood and memory disorder. So they are now taken as a diagnostic indicator to find out if the treatment used for memory impairment and mood disorder is actually active. Now, so this is showing you the normal physiology of the neuron and astrocyte. Astrocyte is showing in green. You see them attaching to the neurons, supporting the neurite growth. All these neurite growths are essential for synaptic formation. See the synapses protecting the neuron. But if there is neurodegeneration and this activities has been impaired, you will see them causing an increase in their expression, trying to mop up the oxidative what's it called? Oxidative damage mediator, basically reactive oxygen species due to the oxidative damaging process induced. Now, so how does astrocyte function in neurocognition? One, it does that by promotion of neurovascular coupling, like connection between the neurons and the endothelial vessels. It helps to maintain uptake of glutamate and GABA via the specific cell membrane receptors for their transport. It also helps to produce antioxidants in oxidative stress induced by oxidative stress. There's a way the astrocyte proliferates. Reactive astrocyte produce its own glutathione that acts that showcase antioxidant potential to ameliorate the destruction of the neuron, but rather protect and repair it. It's also involved in synaptogenesis and helps in developing neural circuits via the secretion of this glial transmitter, which is basically the glutamate and the GABA neurotransmitter within the synaptic cleft. The glutamate is an excitatory neurotransmitter, while the GABA is an inhibitory neurotransmitter. So in case of abnormal proliferation of astrocytes, it shows behavioral deficit. So we see the triband arrangement of the neurons and the astrocyte, showing how it produces the glutamine shuttle for synaptogenesis and the activity. So if there's a decline in NMDA receptor function, it has been attributed to cause memory decline. But overactivation of NMDA receptor function triggers glutaminergic cortical activation. And this overactivation of NMDA receptor is also bad for the neuron. It causes excitotoxicity and damage to the neuron. That's why it is balanced by the influence of GABA and inhibitory tone for this glial transmitter release. So here is showing you a resting astrocyte. When in the presence of an inductor, oxidative stress, it becomes a reactive astrocyte. And if you have a reactive astrocyte, that's a sign of neuroinflammatory process and neurodegeneration. And that one will affect distortion of normal functions of the neuron, resulting in behavioral impairment. So this is another diagram showing you the shuttle between the neurons and astrocyte, aside from glutamate lactate production, and also the shuttle for glutamine release, which is the within the tripartite synapse. Okay, so what are the similarities between the neuropathology of depressive disorder and memory disorder? One is both disorder is characterized by disturbance of the synaptic plasticity, talking about the synaptogenesis or connection between the neurons. We also see enhanced neuroinflammation demonstrated by reactive astrocyte. And when you see reactive astrocyte, is an indicator of neuroinflammation that shows that there's a loss of neuron. And when you have this reactive astrocyte causes increased secretion of all these inflammatory cytokines, interleukins. And this interleukin has been recorded to be like indicator for mood disorder and anxiety disorder and memory disorder. So if there's a drug that has antidepressant effect or memory improving effect, it will act like plays the role of an anti-inflammatory response to reduce this pro-inflammatory secretion from the astrocyte and promote inflammation, thereby reducing the expression of this reactive astrocyte. So based on this tribute of astrocyte, you will see the study over the years that there's an increased report of drugs that target astrocyte as therapeutics for managing depressive disorder and memory disorder. So astrocyte has now been a target for treating memory disorder and depressive disorder because of their effect on the glutaminergic signaling that contributes to increasing brain-derived neurotrophic factors that improve memory. So I've done some studies on models of memory impairment, like using scopolamine to induce memory loss or memory impairment in animal models. And I've seen, like, looking for neuroprotective agents with reported antioxidant property, for instance, alpha lipoic acid, which has been able to reverse memory impairment and all the other neurocognitive impairment demonstrated in this study by targeting the astrocyte. Here also, there was a study that also involves N-acetylcysteine, which is another neuroprotective compound that works by activating intracellular production of glutathione using the precursor cysteine. And this antioxidant activity helps to reverse aluminum-induced neurodegeneration. Aluminum is used to create like models to study Alzheimer's, aluminum is used as the model for Alzheimer's disease. So it also shows that these two antioxidants target the astrocyte by reversing it. Now, currently establishing that there's a correlation between memory loss and depression. The same N-acetylcysteine used to reverse neurodegenerative process in memory impaired model, talking about the Alzheimer's disease, was used to evaluate if it has the same effect for depressive behavior. So N-acetylcysteine also has its ability to reverse depressive behavior by also acting like a neuroprotector, working on the astrocyte and also the synaptophysin by reducing the proliferation of astrocyte, which causes expression of synaptophysin that also helps to alleviate motor dysfunction due to neurodegeneration as well as depressive behavior. So from this report, it's been established that pathogenesis of depression is linked to neuroinflammation and oxidative stress, as seen in memory disorders such as Alzheimer's disease. So further study needs to be done by seeing behavioral effect of antidepressant drugs using forced swim stealth to screen antidepressant drugs. So forced swim stealth is a short study to validate any drug that has antidepressant effect. And all these studies have shown that antidepressant is related to prefrontal cortex hippocampus, as I mentioned earlier. So in this current study, to evaluate antidepressant potential of ketamine on FST models by assessing neurocognitive changes, looking at behavioral changes in the rat, then changes in the histomorphology of the prefrontal cortical neurons and reactive astrocytes, as well as chromatophilic bodies. Using the following objectives, we did H&E stain to establish the neuropathology, also Christopher's valence, and neurobehavioral changes that has to do with immobility time and hyaldonia, because it has been established that antidepressant agents will decline immobility time, and also establish reactive astrocyte, expression of reactive astrocyte, and using GFAP immunohistochemistry, and synaptogenesis using synaptophysin protein. So in this study, 30 rats were used. We have a control group. We have the stress model for the animal model of depression. We now studied using ketamine, 20 milligram per kg intravenous ketamine, and you study it against the standard drug, which is the fluoxetine, fluoxetine has been taken as established in my earlier discussion, like fluoxetine has been used as a drug. Then we now gave ketamine to the FST model as well as fluoxetine. And this study was done according to the guidelines for care and use of laboratory animals and as approved by the Anatomy Department Research Ethics Committee. So we did the FST model by subjecting them to the first swim test. Then afterwards, for the test session to test the antidepressant activity, we gave the drug and allowed the drug to work in the rat for like an hour. Then we now subject them to the first swim test. Then this is how we record the behavior, mobility, and immobility time. Then we also study hardonia. Hardonia is desire for what the animals like to do. So we used the method of sucrose preference test. But we did an habituation test before we did the actual test by studying their volume of sucrose intake and water intake. So this is the timeline. Animals were acclimatized. Then a pre-test, a 15-minute habituation test. Then a 5-minute FST test. Then we now did the actual 5-minute test to test the antidepressant property of ketamine. Then we now harvest the brain and fix the 4% paraformaldehyde to study the H&E of the prefrontal cortex, chromatophilic body, phonetic granules, which is the basis for ribosome, Nissi granules demonstrating ribosome, which is the basis for protein, amino acid building block, or neurotransmitter synthesis. And we check the immunohistochemistry of astrocyte and immunohistochemistry of astrocyte and synaptophysin. So just this study is to establish that astrocyte has a role in cerebral function through neurosynapses and cerebral dysfunction through disruption of neurosynapses that leads to decrease in glutamate uptake and increased release of inflammatory cytokines and mediators. So result of things shows that for our control, there was an increase in brain weight, but the FST model for depression, there was a decrease in brain weight. For the ketamine treated group, there was a increase in brain weight as compared to the fluoxetine treated group. When we now compare between the ketamine treated depressed model and compare with the fluoxetine treated depressed brain model, there was an increase in the brain weight. So this one is showing that supporting report that in stress affects the mass of the brain neuron and the neuropil, thereby leading to decrease in the brain. Neuron atrophy leading to decrease in the brain size. And this atrophy has to do with loss of the degeneration, thinning of the neurites, thinning of the cytoplasmic extension, still the neurites that connect at the synaptic point. Now, any drug that has a potential antidepressant effect any drugs with an antidepressant effect must increase immobility time. So in depressed mood, we see a decrease in an increase in immobility time in the depressed rat model. But in the treated group, in the fluoxetine treated, which is the standard serotonergic system mediating drug, there was a decrease as compared to the depressed rat. But when we now compare it with the ketamine treated drug, there was a significant decrease in immobility time as compared with the depressed model. And when you now compare it with the test drug, with the standard drug of depression working on the serotonergic system, there was a significant decrease in ketamine treated group as compared to the fluoxetine. Now, checking the sucrose consumption, water consumption. Now, sucrose consumption signifies anhedonia. So if there is a decline in sucrose consumption, that signifies anhedonia. Now, sucrose 1, we have two sucrose phases. Sucrose 1 was taken during habituation. Sucrose 2 was taken and in the pre-test. Sucrose 3, the yellow bar, was taken after they have been after the drug and after they have been subjected to the first test, and now exposed to the sucrose solution. So here you will see there was a significant increase in the first sucrose in habituation. Then the second sucrose, there was a significant increase in the control, but there was a decline in the first swim test as compared to the fluoxetine treated that was not subject to the first swim test, and the ketamine treated not subjected to the first swim test. But the drug study group E and F shows that ketamine caused a decline in the first swim procedure as compared with the fluoxetine. But now, after they have been exposed to this antidepressant drug, we found out that the control increased because they are not exposed to the first swim. For those people exposed to the first swim, there was a significant decrease in their sucrose consumption level. Significant decrease in their sucrose consumption level. Fluoxetine decreased. This group was not subjected to the first swim test. So the group we'll be focusing on is group B, subjected to the first swim test, group E, subjected to the first swim test and treated with ketamine, and group F, subjected to the first swim test and treated with ketamine. So in this study, we found out that in those groups, in the antidepressant drug study group, the drugs improve their consumption of sucrose as compared with the consumption of water here. So ketamine treated group prefer to take in sucrose as compared to water when comparing both. Also, their food increase in intake increase. However, the fluoxetine group intake of food increase. The volume of food taken by ketamine group, fluoxetine group increase as much as compared to the ketamine treated group. Now, looking at the neuropathological slide, the control group, we can see clusters of normal neurons in the prefrontal cortex as compared to focal necrosis in the prefrontal cortex of the depressed group. Ketamine treated, you see preserved neuron, no degeneration, preserved neuron. But in E and F, treated with ketamine and fluoxetine respectively, you will see some neuron preserved. However, we still have some focal degeneration in some area, but you can compare it with the group subjected to the first swim test without the antidepressant treatment. Also in the fluoxetine, we can see focal degeneration, but we still have some with normal neuron showing the pointed highlighted with the yellow arrow. Now we establish astrocytes. Astrocytes are used to express neuroinflammation. But in normal cells, we still have astrocytes there just to do their normal function of neuroprotection and synaptogenesis, and protecting the synaptic synaptogenesis. So in the normal, you see expression of astrocytes, but it's not well expressed as compared to the first swim test. So in the first swim test, you see well expressed astrocytes. In the ketamine treated, you see well expressed astrocytes as compared to the control. This one is showing that the ketamine is performing its normal function in the synaptic cleft, preventing reuptake of ketamine at the synaptic junction. So these astrocytes will be there to express to prevent excitotoxicity at the synaptic junction. Fluoxetine is also there expressed because it's also there acting on the fluoxetine is acting on the serotonergic system by preventing reuptake of serotonin. So it's also expressed there to support the neuron activity. But now, see when we compare L, E, and F, group E, which is the ketamine treated, The ketamine treated is showing expression of this astrocyte. This expression of astrocyte is coupled with the presence of ketamine there, trying to speedily cause neural repair, neural regeneration that will crash down the neuroinflammatory process that has been activated by the stress. But you see it expressed here as compared with fluoxetine. So meaning this one has rapid expression rate as compared with fluoxetine. Now, what is the role of synaptophysin in this neurocognitive process in neuropathology? You see, when expressed Brownish deposition of the synaptophysin synaptosome. Synaptosome is the vesicle containing the synaptophysin protein, expressed in immunohistochemistry as brown deposition in the neuropil. So you see them, this well-expressed brown deposition, but this brown deposition is mildly expressed or poorly expressed in the first swim test, showing that there's loss of synaptophysin protein with the increase due to oxidative stress, physiological stress, causing loss of synaptic synaptogenesis, neurodegeneration, losing the integrity of the neuron, loss of neural connection, then loss of synaptic synapse and synaptophysin protein within the synaptic cleft. But here you see the brown deposition in ketamine as also in fluoxetine. You now see the process of reactive astrocyte protecting against neurodegeneration, causing preventing the loss of this protein, synaptophysin protein within the neuropil. We also see the similar thing in fluoxetine. So meaning fluoxetine is showing its own stuff, but it's also more deposited in ketamine as compared to fluoxetine, looking at the brownish deposition. And you will still see, you can see some dead neurons there, but they are still protected neurons as seen there. So there's a correlation between activities of astrocyte as per maintaining neurocognitive activity and neurocognition in the brain. So we can now say that from this study, this is being deduced that means rapid antidepressant potential is demonstrated in its ability to reverse neuroinflammatory process. And as it reverses that neuroinflammatory process due to the activities of astrocyte, it also helps to increase synaptogenesis and synaptic connection. And this synaptic connection is the brain block that helps to improve neurocognition and alleviate depressive-like behaviors. Hence, ketamine has demonstrated its potential also to become a neuroprotective agent that can reverse neuroinflammatory associated with memory impairment through its ability to improve astrocyte neuron interaction and synaptogenesis for memory potentiation. So these are few of my references. Thank you for listening. I want to appreciate my university for their support and the organizer of this live conference for the opportunity to present this work and discuss and establish that there's a correlation between the neuropathology of dementia and depressive disorder. And for both therapeutic, for both of them, their therapeutic agent and their diagnostic tool is to establish astrocyte proliferation as well as expression of synaptophysin. So thank you very much.